This will be a rather short discussion. Just want to reemphasize and make clear that in your heads you can distinguish what the difference is between a motif and a protein domain. Now remember, exons roughly correspond to the functional protein domains in many cases. It's not a 100% rule, but it's a very, very clear pattern that we see in the eukaryotes. And sometimes the term motif is used interchangeably or maybe not as carefully as it should be. So I want to be clear how we are going to use the term, and I think this is the most logically consistent way to define this, but just be aware there might be a little bit of flexibility in how some people use these terms. So a motif is any short section, well, it could be short to long, but any section of DNA that is conserved across different organisms, and so it is recognizable. And so we could have a promoter region, for instance, that we see at the front of genes that is much the same across many, many dozens of species, and that would be a motif. We could have a region within the protein itself that is very conserved as I look at 10 or 12 or more species and see that short section. And so motif does not have to correspond to any sort of functional unit of a protein. Motif is simply a conserved recognizable sequence at the DNA level. And if it's in a protein coding region, of course, if you can recognize it at the DNA level, you're going to be able to recognize it at the amino acid level. Okay? And so just like exons and exon shuffling, these motifs can be duplicated. Now, the little bit of confusion, I think, comes from really a kind of an idea of scale rather than complete difference between what a motif and a domain is. Really, you could think of a motif as a more inclusive word or definition that includes little short sections that are conserved and also really, really long sections, everything up to complete domains, and that could be a motif. But usually the term motif is used for much smaller regions, subcategories or subparts of an overall domain or a small regulatory region or something like that. And so via the same mechanisms, unequal crossing over, we can get duplication of these motifs. So we see them over and over and over again in some genes. And some genes are notorious for this. Many immune response genes in mammals have these little regions that are repeated many, many, many times that give them slightly different properties. And so the same mechanisms that cause exon shuffling and duplication of domains via exon shuffling can also cause duplication of motifs, but it's on a little bit of a smaller scale. So we can do this via ectopic recombination, right? Unequal crossing over. But another common mechanism, and it's more common for motif duplication because of the scale, would be a slippage of the DNA polymerase during the replication phase of cell division. And so that would represent a mutation. We might have 2 or 3 instead of 1 repeated motif, and we'd see that, and that would add diversity. Now again, it's a mutation, just like an exon shuffle. Most of the time it's probably not going to be beneficial, going to be negative, going to be detrimental, will be removed by natural selection. But enough of the time it results in something positive. And so this is the major mechanism, this copied with gene duplication. So really, we're looking at scales of different things, but gene duplication, exon shuffling, motif duplication, these account for the vast, vast majority of all of the diversification of proteins, especially in eukaryotes, but to some extent even in the prokaryotes. Rather than just having random mutations create a working protein from scratch, from non-coding DNA, copying what is already there and then making modifications of that by random mutation and natural selection, acting on that random mutation, is a much more common and likely scenario. In fact, it's probably very rare over the course of the history of life on Earth to have protein evolution de novo from nothing or from non-coding DNA. But new proteins evolving from duplicated genes, or a new protein that's kind of a Frankenstein version of an older one, those are very, very common things that occur across the history of life on Earth. But they are still just new mutations. We would need to evaluate each one individually as to whether it was positive, benefit, or neutral. Because they are in coding regions and have such a major change on the genome, they are probably most of the time they're in that small subcategory of genes that almost always will have a phenotypic impact. Maybe every once in a while one would be neutral, but most of them are going to have a phenotypic impact. So probably most of those, maybe 95% of all of them are going to be negative, but a small percentage are going to be positive and kept. And then those are the ones that perpetuate, are selected for by natural selection, and create the diversity of the protein repertoires that we see today. So we can recognize them both at the DNA level and at the amino acid level. And occasionally you will see a motif that is represented like this. And this is over perhaps maybe a dozen or more different species. And they're looking at this one little small helix turn helix motif in a family of proteins. And so what they've done here is they have put a letter representing one of the 20 amino acids. G, I think, is glycine. Arginine is R. I don't know all of them. Serine, I think, is S. But whatever the 20 letters represent, the size of them, how large they are on the scale, represents how many of the species have it. So maybe only 1 out of 20 has whatever that smashed thing is supposed to be. And the vast majority of them have G. And so you can see some of the amino acid residues are much more conserved than others, which might show some conservation. And then others are very, very flexible and have 8 or more different possible amino acids that occur there across the sample size. Okay, and so this is just a way to visually represent the diversity across a motif and still show that there is remarkable conservation. Now, when we see this, this means that there is strong natural selection to maintain this sequence of amino acids in the organism. And if we looked at this at the DNA level, we could do the same thing, but with just A's, T's, C's, and G's to represent the nucleic acids instead of the 20 letters to represent the amino acids. Okay, and what we would see is probably more of a pattern where the second and first positions would be very conserved, especially in these areas where we have very little amino acid diversity, and the third positions would just be all 4 of the above. Okay, where we're going to have more diversity, and that just has to do, of course, with the DNA code. So anytime you're looking at a motif, you could recognize it at the DNA level, but it's going to be more conserved at the protein level if it is in a protein coding sequence. However, not all motifs need to be in protein coding sequence. Because we are defining a motif just strictly by its conservation of sequence, we can define them at the DNA level. And so if we see a motif at the DNA level, that means that there's been some strong natural selection to maintain that sequence at the DNA level, even though it may not be in the middle of a protein. So natural selection is not only for proteins. And we'll look at an example here at the end of this discussion. So motif duplication also has to do with alternative splicing. There are some genes that have evolved to have motifs or domains, and again, depending on the size of these, we may define them differently. Some of these might correspond to an entire functional unit. Others might just correspond to a small secondary structure that's not really a complete functional unit. But some genes have evolved to be spliced together in multiple ways. Major histocompatibility genes in mammals are one example of this, where there is multiple motifs that are duplicated and then alternative splicing mechanisms that can make 3 or 4 or 5 or sometimes dozens of final complete mRNA transcripts from the original gene itself. So this is a gene, but with 3 different protein products that all have slightly different properties. And so this can be very important where diversity of outcome is important. And that's not a super common thing, right? Because mostly you want a conserved result that always can do the function it's supposed to do very, very well. But there are some biological functions that are well served by diversity. And the major histocompatibility genes are responsible for helping to recognize antigens, these, bacteria, viruses, fungi, whatever gets into a mammal, and our immune system needs to be able to recognize it. So having a diversity of different combinations that can bind differentially to these antigens has been a huge advantage and one of the major trends that we see in the evolution of the mammalian immune system. And we'll talk about that a little bit more briefly when we look at the Pagan paper. I'll just do a quick review of it in the next lecture. Okay, And so promoters and regulatory regions are non-coding parts of the genome that can have very important motifs, and we can recognize them graphically here also. These are, I guess, different looks at similar motifs across different ranges of organisms. And some, it's very, very conserved, so some areas are very conserved. This may be a promoter. I think this one's a promoter. But others might be a regulatory region that's really important for a binding site of a transcription factor for proper expression of a nearby gene. Okay, so again, in your head, just make sure you know what a motif is, this slight difference and important distinction between a motif and a domain.